In this video, we are going to discuss the relative advantages and disadvantages of gateway cloning. The pros and cons of gateway cloning are summarized here. We will discuss them one at a time. The first advantage to discuss with regard to gateway cloning is its relative speed. Gateway is fast. The BP and LR reactions are considered complete in 5 minutes. In addition, the entire system relies on only 2 enzyme mixtures. This makes the system very high throughput. One entry clone can easily be adapted by transferring to different destination vectors. Let's say that you have cloned a novel protein from mouse. You may want to express the protein in bulk in E. coli. Or you may want to screen for proteins that could interact with your novel protein using yeast 2 hybrid screening. Alternatively, you may want to make a fluorescent fusion protein. Or you may wish to do all 3. After you determine which destination vectors suit your objectives, you are 3 different LR reactions away from 3 significant and useful experiments. There is no need for repeat sequencing. Once you have sequence verified your entry clone, you can be confident it will be the same as you transfer it to different destination vectors. Depending on other components in the destination vector, you may wish to sequence across junctions, but the sequence of the gene of interest should not be affected by the BP or LR reactions. Gateway makes multiple fragment assembly easy. The gateway system actually offers 5 different variants of the @B and P sites with their corresponding @L and R sites for cloning. These are generally referred to by number, for example, @B1, @B2, etc. These sites differ from each other only by a few nucleotides, but allow sequence-specific BP to LR transitions. Taking advantage of the sequence specificity of these 5 classes of sites, multi-site gateway cloning can be used to assemble 2, 3, or 4 inserts into the appropriate destination vector in a single reaction. No doubt the first and most obvious con of gateway cloning is that it inevitably leaves scars. The @B, P, or LR sites are not eliminated during any of the cloning steps. The next disadvantage of the gateway system is that extra care must be given to storage and handling of the reagents. All of the gateway vectors contain a copy of the CCDB gene, which kills the most common E. coli strains used for cloning. This is a safeguard in the technique to select for recombinants. However, in order to propagate any vectors in your lab, you must be sure to use CCDB survival cells. Also, unlike restriction enzymes, which are generally stored at -20 degrees Celsius, the BP and LR clonase enzymes should be stored at -80 degrees. The product literature suggests -20 or -80 is okay, but anecdotal evidence suggests storage at -80 degrees extends the shelf life and the activity of the enzyme. In principle, the gateway cloning reactions are not subject to background due to the incorporation of the CCDB survival gene. In reality, there is a risk of background colony growth if the CCDB survival gene acquires enough mutations. Standard transformation protocols suggest a 1-hour recovery incubation after heat shock. Some researchers have found that this full hour increases background, apparently from spontaneous loss or mutation of the CCDB gene. Background colonies can be minimized when you reduce the length of the recovery period after heat shock. The gateway cloning vectors and the associated BP and LR clonase enzyme mixtures are relatively expensive. The entire system is proprietary to the Invitrogen arm of Thermo Fisher, and there is no direct competitor on the market. It can also be somewhat labor-intensive to create all the entry clones that you need for your experimental system. In the event where the cassette-like nature of gateway cloning benefits your experiment, the investment to establish the reagents is well worth the effort. Here is a reminder of the pros and cons of gateway cloning. To learn more about molecular cloning, visit SnapGene.com.